This video abstract is AI generated. What if the best way to inhibit FTO is not to target the active site, but the moving interface that holds the enzyme together? FTO is a human RNA demethylase that removes M6 from RNA. Because FTO activity is linked to RNA regulation, obesity, and several cancers, the enzyme has become an important target for inhibitor development. But many FTO inhibitors still lack potency or selectivity. FTO contains two domains. The N-terminal domain contains the catalytic machinery for metal, alpha-ketoglutarate, and substrate binding. The C-terminal domain does not directly bind the substrates, but it is required for proper enzyme function. This raised a central question. What role does the C-terminal domain play, and what does APO-FTO look like in solution? Solution NMR provided the first answer. Full-length FTO gives a well-dispersed NMR spectrum, consistent with a folded protein. In contrast, the isolated N-terminal and C-terminal domains give poorer spectra with fewer, broader signals. This shows that the interaction between the two domains is required to stabilize the catalytic domain in its functional conformation. The next question was whether a static crystal structure fully describes APO-FTO in solution. Residual dipolar couplings measured by NMR did not agree with the crystal structure, indicating that APO-FTO samples a dynamic conformational ensemble rather than one rigid structure. NMR relaxation experiments confirmed this picture. FTO undergoes motions on both the picosecond to nanosecond and microsecond to millisecond timescales. These dynamics are especially prominent in peripheral loops, near the active site, and at the interface between the N and C terminal domains. To visualize this behavior, NMR residual dipolar couplings were combined with accelerated molecular dynamics to generate a 39-member solution ensemble. This ensemble satisfies the experimental NMR data and reveals that the interdomain interface is more flexible in solution than in the crystal structure. Most importantly, this flexibility creates transient surface pockets at the interface between the N and C terminal domains. These pockets are not visible in static crystal structures, but they appear in the solution ensemble and are predicted to be druggable. One pocket overlaps with a known selective inhibitor binding site. Together, these results show that FTO function depends on a dynamic interdomain interface that stabilizes catalysis and opens transient allosteric pockets. These pockets provide a promising route for designing selective FTO inhibitors. To know more, read our work online.